Traditional knowledge of medicinal mushroom, including Antrodia cinnamomea, 牛樟芝 to intervene subhealth status in humans。我大概把它翻译是要用乎传统知识的价值，包括牛樟芝对人类亚健康状态的影响。Let's welcome.
Good afternoon. I will be speaking in English. I hope I don't go too fast and hope to keep my time. This morning we had a very good lecture by our keynote speaker who told us a lot about fungi, the macrofungi, and all the wonderful things that we get from the mushroom. The earlier speaker actually gave a very good talk because many a time we are presenting the goodness of mushroom. We are working with extracts or some enriched extracts, and we say these compounds are present, but we don't know what happens the moment we take it. All right? I think the last study is a very good study. I think many of us should be doing that before we can really claim medicinal mushrooms are good. This afternoon, I actually want to share with you why we should be looking at traditional knowledge. The very fact that Taiwan is investigating Antrodia cinnamomea is based on traditional knowledge. And here I want to touch on something known as sub-health status. We have looked at a number of papers looking at liver cancer, liver protection, neuroprotection, right, and also cancer. Some of these are actually prevention, chemo prevention. Some are heading towards treatment. But here we are going to look at the sub-health status in humans. I'm from Malaysia. And if time permits, at the end of this lecture, I'll tell you a little bit more where I come from. In the meantime, if I run short of time, I would like to record my thanks to the Institute of Biological Chemistry, Academia Sinica, and Professor Chen, and especially the Taiwan Edible Medicinal Mushroom Fungi Biotech Association and the Taiwan Federation of the Antrodia cinnamomea industry for my being here, standing in front of you, trying to share some of our research that we do in Malaysia. So what is this sub-health intervention? Sub-health intervention is trying to prevent what we know as a full-blown disease. Now we actually face a number of diseases which are life-threatening. We talked about cancer, we talked about cardiovascular diseases, and there are also like diabetes, and also the neurodegenerative diseases. As we age, we seem to lose our memory, and that can be very, very traumatic. So what we are trying to do is, we're trying to prevent rather than cure these life-threatening diseases by reverting to traditional knowledge as a source of chemo-preventive food and nutraceutical. And mushrooms seem to have center stage because in many cases they are used by the traditional communities to stay healthy, to prevent disease, and in some certain cases also to treat disease. So what we are trying to do is to use mushrooms so that we don't end up having cardiovascular hypertension or diabetes, or end up losing our memories at an early age so that we can have better quality of life. And this sub-health condition is what we normally call those people maybe in the 40s to the 50s, where we having pre-diabetes, pre-hypertension. That means all our biochemical markers like cholesterol and other markers are all what they say slightly higher than normal. Slightly higher, maybe the doctor don't want to give you medicine, but definitely, slowly, we will end up having to take medicine. So why traditional knowledge? I think mushrooms have been a very long fascination for many of us. If you have fairy tales, I'm sure in the Chinese history and the Taiwan history also you have fairy tales. This is very popular. This is Alice in Wonderland. How many of you have seen the movie? Have you all seen the movie? Yes, I'm sure. So you find this, they are dancing around mushrooms. So mushroom figures very much in fairy tales. They also have been part of immortality in a number of rituals, and one of the mushrooms that is used here is the Amanita muscaria, very beautiful mushroom. It is used in traditional, what we call as rituals by the medicine man or the shamans. Mushrooms also have healing properties. All right, we have visionary mushrooms throughout the world. So you find that in many cultures, mushrooms are part of their caricature, part of their writings. All right, so mushrooms are also featured here. So it's been part and parcel of mankind for a very long time. If we talk about the Iceman, he also had a couple of mushrooms in his bag, the Piptoporus and the Pomace. So when they found the first Iceman, what did they find in the pouch that he had round his waist? Were 3 fungal species. All right, this is the one that I showed you just now, the Pomace and the Piptoporus species. So it is not something very new. Mushroom is quite old. Of course, in mainland China and this part of the world, culinary and medicinal mushrooms are very much part of the ethnomycological information in China, Japan. So we, and especially you folks, have very long time been associated with mushroom, especially this one known as the reishi mushroom or the lindsay. So what is that in the mushrooms and that gives so much of its properties? As mentioned earlier this morning, I think we can get the medicinal properties from fruiting bodies, submerged cultivated mycelia biomass, as well as the liquid broth are the sources. The majority comes from the fruiting bodies. Some amount comes from the mycelium, and we can also get from the broth. That means the liquid part, because the mushroom excretes these bioactive compounds into the liquid medium. So these are some examples. I'm not going to go through all the different types of mushroom, Phalinus, Lanthinus, Edodes. These are some of the medicinal properties, or I'm sorry, compound. Caffeic acid, cordyceps is cordycepin. Then we have the Pannus, which is a cyclooxidone, and even the Lanthinus, they have some compound. Sorry, I'm not a chemist, so I have a tongue twist with all the names. Right? Now, what are the activities of these mushrooms? Many of them have anti-tumor, hepatoprotective, we heard about it, anti-diabetic, cardiovascular health, and the one that I'm going to talk to today is the brain and neuronal health. The main functions of these mushrooms or their properties are due to some basic functions like immunomodulatory, anti-inflammatory, antioxidant. Some of them protect your DNA and many of them can protect your cell. Now I'm going to talk mainly about brain and neuronal health today. Now there are many mushrooms that are responsible, but before that I just want to go through some process. Now the most important part in nerve health will be neuritogenesis. This is a process when the cell of the nerve cell generates elongated axon and dendrites. And this is very important and is in 3 stages. First, the neurite is initiated from the neuronal cell. Then the neuronal cell will elongate and maintain the structure, the integrity. and finally it will form a network. Now when all these things happen, then we have memory, because all the nerve cells have to connect. Now what happens is, as we age, senescence, as we age, the neurons seem to die. And this is the most important factor in age-related neurodegenerative disease. That means as we age, we will tend to lose our neuronal cell. One of the reasons is because we lose this chemical called neurotrophin, and that reduces our brain cell function. That's why as we age, we seem to lose our cell and also our memory. So this is the nerve growth factor. Now the nerve growth factor has potent biological activities. It promotes neuronal survival and neurogenesis. It has been targeted as a potential therapeutic drug for the treatment, possible treatment of neurodegenerative disorders. But what happens is NGF is very unstable and it cannot cross the blood-brain barrier, what we call BBB. It has to cross from the blood into the brain cells, and there's a barrier in our brains, and because NGF is a high molecular weight polypeptide, so it cannot go through. So we have to look for other things that can stimulate NGF production, or it can behave like NGF. Now among the mushrooms I've selected Hericium. I did not select Ganoderma. I particularly selected this one because I have a story to tell about this mushroom. This Hericium erinaceus is a temperate mushroom. I think you call it the monkey head mushroom in this part of the world. It's quite tasty, nice to eat, and a lot of research has been done. So it's traditionally known mushroom, mainly for gastric ulcer purposes, but also to increase stamina and also the general well-being of the human. And a lot of work has been done, a lot of compounds have been identified, and among them is the erinacins from the mycelium, which are supposed to be strong stimulators of nerve growth factor synthesis. Remember, I told you NGF is very important for neurogenesis. So because of that, they say potential in Alzheimer, anti-dementia, and in our work, we showed that it is also good for peripheral nerve regeneration. Now, I'm not giving you all the results, but if any of you are interested, please email me and I can send you a lot of the papers. What we did is this temperate mushroom can now be grown in Malaysia, which is a hot country. We have high temperatures up to 35, sometimes 32 to 35. And at the same time, that property which stimulates the neurite to grow is maintained, though grown at high temperatures. So we have published a series of papers done by my PhD and master students. So if any of you are interested, then you can write to me. So in this case, we have reached up to the animal world. So maybe now we have to look at the pharmacokinetics and see how actually what happens when we consume this mushroom. Will it really help our brain keep on producing NGF so that we will not have this age-related memory loss at a rather early age? Now the other mushroom I would like to talk about is the Lignosus rhinoceretis, which is known as the tiger milk mushroom. Alright, in mainland China, there's also another mushroom which they call as Pleurotus tuberigum. It looks like that. Here the cap looks like the Ganoderma cap, but what is important is the sclerotium. It's like a bulb. This is under the ground, and this is the one that has packed with all the medicinal value. So it is found in all these parts. We say they're found in southern China, in parts of Thailand, Indonesia, and also Philippines, and Papua New Guinea, and as well as mushroom. This mushroom has been described. This is one of them. We heard there are a few more, and there have been publications recently for many other species, a couple of other species. Now, what is this mushroom? This is a traditionally used mushroom, and it has been documented by Chang and Lee et al. That the sclerotium has many medicinal properties. It has been used by the indigenous communities to treat fever, cough, asthma, which is an inflammatory disease, related disease, chronic hepatitis, gastric ulcer, and even cancer. All right? So in cancer, mainly for breast cancer. So there's a lot of research now ongoing in many universities, also including in Hong Kong. So what has been reported and published is anti-tumor activity, immunomodulatory activity, anti-inflammatory activity, antioxidant activity, and in our own group, we have shown that it stimulated neurogenesis. Now, why did we look at neurogenesis, though the ethnopharmacological document or the oral history did not show this information? But when we talk to the indigenous people, they take this mushroom as a brew, that means they boil it, and they drink it and before they go hunting. Because they say they want to remain alert, they want to remain active while they're hunting deep in the jungle. So we believe it has something to do with our brain activity. So we decided to look at it. So when we did at the different extracts, hot water extract, ethanol extract, and crude polysaccharide, we found that the crude polysaccharides were not so good, the ethanolic extracts were good, and hot extract was good, very good. Compared to, this is a negative cell control, which is only the cell, and this is 50 nanograms of NGF, or the nerve growth factor, commercially available. So what you can see is these thin lines here by neurofilament staining confirms that these neuronal cells that you see here, but no neurite, and these with NGF stimulated have neurite, is because we use the PC12 cell line. And PC12 cell line, for neuroprotection, it's fine, we can use a lot of chemicals, but if you want the neurites to grow out from these cells, you must have NGF. So when we have NGF, you get good neurite extension. When we add our extracts, again we see very good neuronal extension, which is confirmed by the NF200 antibody staining that we do. Okay? So we try to map the pathway. We believe that the nerve growth factor activates the extracellular, what you call tyrosinase kinase, like an A, which has an affinity for NGF, and that will trigger the RAS-RAF pathway through the mitogen activator kinase pathway and the extracellular signal regulator kinase, and finally neurogenesis will take place. So you have NGF, it will go down this pathway. Now when we use inhibitors, we try to use pharmacologically available inhibitors, either block at the activation, or further down the line where the MEK pathway will move down to ERK and finally to neurogenesis. Because we believe this pathway is needed for NGF and hot aqueous extract for promoting neurogenesis. And what we found is true: when we block at these very critical points, neurogenesis doesn't take place. Now what we also found, this is what we see, so the inhibitors block the neurogenesis activity. What we found is something very interesting. When NGF, when you add the inhibitors, definitely the pathway is blocked. But when we added the aqueous extract, when we have the inhibitors, again we find there is neurogenesis, but there is no NGF production. So this tells us that the activation is necessary. That means MEK ERK pathway signaling is necessary in PC12 cells by NGF. And here we found that the hot aqueous extract mimicked the NGF. That means the NGF levels did not go up. That means we did not add NGF to those that had the extracts, but still the neurogenesis took place. So here, most probably in this mushroom, there are compounds which mimic the NGF activity. This is an agreement. There are many studies for Ganoderma, Ricola frondosa, Pleurotus giganteus, and this Pleurotus Ganoderma species from our own group, where we have shown that this pathway is essential for neurogenesis, and this is the pathway that is involved in mushroom-induced neurogenesis in PC12 cell lines. Now, when I was invited for this meeting, I told Mr. Brad, who came from the association, this is an Antrodia meeting. I don't work on Antrodia for the simple reason we don't get this mushroom in Malaysia. Then he said, why don't you come and present some of the other work you're doing on traditional mushroom? I said, fine. And then we had a discussion, and somewhere along the line, he agreed to send me some samples. All right, and we did some work. Very preliminary over the last 1 month. So it's just like you're topping on your ice cream. Where we want to go from here depends on the people who are interested in this fungus in this country. So what we did the neuroid outgrowth activity of Antrodia cinnamomea. We used PC12. I saw that there are publications with Antrodia and PC12 cells, mainly for protection, against inflammation, against protection, but they did not use PC12 for neurogenesis, and that's what we showed. So we looked at, they gave us a number of specimens, but finally we used the vesicular carp, the fruiting body, what they call membrane, and what they call mycelium. After we came here on Sunday, we really clarified what all these things were, and there are some interesting results. What we found that PC12 cell line, this is NGF at 50 nanograms. The control is just our cell line with no addition of NGF, so there will not be much neurite-bearing cells. The percentage is very slow. But when we use the mycelium, which I believe comes from the solid substrate fermentation, and then the vesicular carp, we found that neurogenesis did occur. There was neurite outgrowth, which was even slightly better, but not significantly better than NGF at 50 nanograms. So there are components in Antrodia which can help in neurogenesis. Similarly, when we measured, that's where we saw, although the percentage number of cells producing the exon are not significantly different, but in terms of length, the mycelium outshone the vesicular carp, which was comparable to the NGF at 50 nanograms, but the mycelium was significantly higher at P0.05. So this is very preliminary result. So there's a lot of scope to investigate this neurogenesis process, which is very important, especially when our nerve cells are dying. When they're dying, actually what happens? The neurites are not extending anymore. So when the connections are not met, then we become forgetful. All right? So these are the neuronal cells. All right, we have not done the, what we call the neurochemistry. We did not do the filament staining, but definitely we can see that this is the control, and B is the one with the NGF, the PC12 cells, need them. And if you can look at C, which is the mycelium and the vesicular carp, you can actually see very good elongation of the neurite. So definitely this mushroom supports neurogenesis. We can explore further looking at the pathway and even at other possible use for this mushroom. Now we have a poster on the antioxidant activity and cytoprotection by my student who looked at it against glucose oxidase induced oxidative stress. This is more looking at the potential in diabetic, anti-diabetic, or to reduce incidences of diabetic through a chemopreventive measure. Please visit the poster. Now we did some anti-inflammatory work because this is ongoing, and we did not, I will not go into this result because I saw a number of presentations and also a number of posters. Again, we used the raw cells. It did not have effect on the viability except at high doses. This is the mesothelioma, the membrane, and the mycelium. The nitric oxide production was of course reduced by the mesothelioma, not so much by the other components, but as we increase the concentration, more nitric oxide was formed, and this increased nitric oxide in LPS-stimulated raw cell was actually due to the cell death, nothing to do with the mushroom triggering the anode production. Alright, so I will stop here, and I would like to thank the organizers, Academia Sinica, as well as the Taiwan Edible and Medicinal Mushroom, Fungal Biotech Association, Taiwan Federation of the Antrodia Industry, all my students, and all those that the grants that supported our work. Now I can take 2 minutes, 3 minutes. I just want to tell a little bit about where I come from, the Faculty of Science. We have a Mushroom Research Center headed by us, and we have 12 principal investigators and about 12 different associate members. We have a bioresource unit looking at the biodiversity of different mushrooms. And then we actually go out, we collect, and then we make the culture collection. And we do this because we need them for all the research that we do, including the medicinal properties. So we have about 1,000 herbarium specimens and about 500 live cultures at the moment. We have the biotech unit where we do most of the work that I just presented. Also, we look at domestication of wild and temperate mushrooms, and then the characterization of enzymes and secondary metabolites. All right, basically what we are trying to do is to exploit our resources as sustainably as we can, acquire the diversity so that we can do the research, and finally we want to take the findings to the consumers, the mushroom growers, and this we do together with the Agricultural Department and the association, the Mushroom Research Association of Malaysia. Thank you very much for your attention.